So, ladies and gentlemen, and Mr. Chairman, first of all, I'd like to say that I'm very grateful to have been invited to speak here today. It's a pleasure. Thank you. And I'm going to talk about the significance of gut microbiota in early age. And these are all potential sources of bacteria for colonizing the infantile gut, and to different extent, depending on the circumstances around the birth and after. But the composition of the early microbiota, the very early microbiota, differs between infants. And in fact, it differs more very early on than it tends to do after, a little bit later, after 1 month or 2 months. And the composition of the microbiota, the early microbiota and the microbiota in general, we all agree that is very important. And I think that it's sailing up as more important every day because this list is not at all complete. I know that because it's added to it very, very often. But allergy has been on this list for quite a while, and we have been thinking that the microbiota, perhaps specifically the early microbiota, would be of importance for the shaping of the immune system, for the programming of the immune system, and so that we could avoid allergy. And I think that most of you is familiar to the hygiene hypothesis. And I can tell you that in Sweden, everyone is familiar with the hygiene hypothesis. And that is because of that researchers has been out and about in the press and talking a lot about it. And one of these researchers is Agnes Wold. But even though we in Sweden, as I say, we know about the hygiene hypothesis, but it was an Englishman that was launching it from the beginning. It was David Strachan that was writing in 1989 his article about hay fever hygiene and household size. Around 10 years later, Agnes Wold, that is a professor at Gothenburg University in Sweden, and as I said, she has been talking a lot about it to the society, communicating it so that everyone knows. She wrote her own version of the hygiene hypothesis that she was calling the hygiene hypothesis revised. Is the rising frequency of allergy due to changes in the intestinal flora? And that was done in 1998. And that was, in fact, what was the background to a project that was run by her and by David Strachan, by Paolo Marticardi and myself during 4 years between 2002 and 2006. And as you see here, we were sampling altogether 300 infants at 6 different time points. And then the end point of this study was that we were looking at allergy in the infants at 1.5 years of age. And this was a culture-dependent study, and we were actually aiming at finding the missing bacteria in allergy. But as you see here, you can see that we were kind of disappointed because this study does not support the hypothesis that sensitization to foods or atopic eczema in European infants in early life is associated with lack of any particular culturable intestinal commensal bacteria. Very disappointing. But we saw differences in this study to what has been seen before 1990. If we compare here, we can at least see that it seems that we have less contribution from vaginal flora and also from gut flora. And we can also see that we have more contribution from skin flora. And especially low was the lactobacillus percentage at 1 week of age. It was only 12% of the kids that was where we could grow lactobacilli from, I would say. So, and here we were 3 years into the study, 1 year left, no significant findings, but 300 very well-characterized children. So we were kind of panicking, I would say, and we decided to do it the other way around. We chose 35 well-characterized infants, and 20 of those were altogether healthy, and 15 were altogether atopic at 1.5 years. They had eczema, they had high IgE, and they had specific IgE. And we were looking at their fecal samples at their age of 1 week. And yeah, they had atopic eczema or not. And we used T-RFLP that was based on the 16S RNA gene to look at these fecal samples. And as you see here, we found a reduced diversity in infants who developed atopic eczema. So the kids that had eczema, atopic eczema at 1.5 years, they had less peaks in their patterns after terminal restriction fragment length polymorphism. And we were very happy, of course, that we had all these well-characterized samples, that we had a method that turned out to be very good when it comes to look at diversity, and that we also got this nice result at the end of this project. And even more happy, we have become even more happy afterwards, because many other studies have actually been showing the same. So have they, even if they have been using other methods, and even in much larger cohorts. So I think that the obvious question now is, why do some kids have lower diversity than others? And a reduced diversity has been seen in other studies. For instance, in this very nice study of colic children, colic infants, compared to control infants, it was seen that the colic infants had a lower diversity, less diversity than the control infants when they were 14 and 28 days old. And as you can see here, the early differences in the composition of the flora was that the colic infants had a higher relative abundance of proteobacteria at 14 days. And these were the differences that was seen early on in this study. And the control infants, they had a higher relative abundance of bifidobacterium and lactobacilli at 7 days and 14 days, respectively. Another study where they also saw reduced diversity was done on very, very early fecal samples, the meconium samples. And there they could see or divide the microbiota in two types, they say, in a type A microbiota and a type B microbiota, dominated by different families of bacteria. And the type A microbiota that was dominated by the Enterobacteriaceae family, Enterobacteriaceae, also turned out to have a lower diversity than the type B microbiota. We have also done a study on the very, very early fecal samples, the meconium. It was done because we wanted to really see, to get a look into the pioneer gut microbiota, because we were a little bit puzzled about the fact that we only found 12% of the kids having lactobacilli in the allergy flora study, as I showed you. So we wanted to see what it looked like earlier on in terms of that. And then we also wanted to see if there was any difference in the gut microbiota, this early one, depending on the birth weight of the infants. So it turned out to be a study called the Pioneer Gut Microbiota in Human Innards, vaginally born at term. So all these kids, they were vaginally born at term and no complications, and the fecal samples were taken within 48 hours, and they were all healthy. They were all at what we call a patient hotel in Malmö, and they are not allowed to go there if they have any complications. So it's a very, very well characterized cohort, actually. So this time we were using qPCR to look for different bacterial groups or bacteria in the fecal samples, and as you see here, all the infants had lactobacilli in their fecal samples. And now we can't, of course, compare with the grown cultured samples, because these lactobacilli could as well be dead. But anyway, they are there, or they have been there living. And we also saw that the other ones with a little bit low enterococci, perhaps, we thought, but otherwise it was nothing remarkable. But the lactobacilli was there in all the kids. And then the question about if birth weight had any relation to what we found in terms of microbiota. We had in this cohort 10 infants that were born large for gestational age. And the other 69 was born appropriate for gestational age. And it turned out that there was actually differences in the gut microbiota of babies born large or appropriate. It was more gram-negatives in the large children, and more gram-positives in the appropriate. And also there was more proteobacteria in the large for gestational age, and more Firmicutes to be at the division level of bacteria taxonomy in the appropriate born for gestational age. So we wanted to look a little bit closer, and we were cloning and sequencing 5 of the large and 5 of the appropriate. And as you see, there was more E. coli in the large for gestational age. And it seems to be a higher diversity, even if it's not significant, because it was too few libraries to get significance. So I think that you have been reading between the lines here that we are interested in the significance of the dominance of E. coli early on, and what that is doing to the host. So we have been addressing that, and now we are talking rats, of course. We have addressed that question in a rat model that we have been working with for a while. And so the question here, what effects do the maternal diet have on offspring physiology? And I'm really the first to regret that we were not looking at microbiota in this study, or not looking at the diversity. But we were looking, together with two physiologists that we did this study with, on a lot of other hard endpoints, I would say. So this was a study that was called Effects of a High-Fat Diet During Pregnancy and Lactation is Modulated by E. coli in the rat offspring. And this was the study design. We were mating the rats, and 1 week after they were separated, the dams were separated and given different treatment for 4 weeks. So they got it until parturition, and then during lactation for 2 weeks, and then we took the pups to look at them. And they either got low-fat diet, high-energy diet, or high-energy plus E. coli. And remember now, it's given to the dams. So, and what happens to the pups when they were 14 days? Well, pups eating E. coli had a higher body weight. They had more fat. They had more, the weight of the liver was higher, and they had more haptoglobin in their blood. And haptoglobin is acute phase protein that is made in the liver upon inflammation. Moreover, their spleen were heavier. And finally, the permeability, the paracellular permeability was higher in the pups whose mothers has been given E. coli. So the maternal diet and E. coli consumption affected the offsprings for sure. Coming back to diversity, that I wish really we had been looking at in that previous study. I think that we have to consider all these sources of gut flora to the infant in order to get hold of the reduced diversity. We have to consider them so that we don't reduce diversity more, but we have to increase the diversity. And it could be very easy. It might be very easy if you consider that you could, just by giving a pacifier to an infant and that you are putting in your mouth once in a while, you could protect against allergy development. That was the earliest news from the Gothenburg group. So I'm sorry if I have been putting up more questions and answers about the significance of the gut microbiota. Thank you for listening. And I would also like to thank Caroline Carlson that has provided me with a nice pictures in this presentation, some of them. And I'm also looking very much forward to listen to the speaks that will be after tea. Thank you.
Questions from the floor?
I have a question. We've looked for the Lactobacillus vaginal species that we find in women, in infant, and we cannot find them in their gut. So why is, I mean, do you have any idea why vaginal delivery is so, is good for?
So when did you look at what age of the kids?
We looked at, we have a cohort for celiac disease that we follow over time. So we looked at every stage and, you know, we can't find them. You can't find Lactobacillus cereus, crispatus, and so you find Lactobacillus, plenty of them.
In this allergy flora, we were looking very thoroughly at the lactobacilli. So we were looking actually at the species level and also at the strain level in all the kids, actually, in all the 300 kids at all time points. You could imagine. and what we saw was that very early, like at 1 week, we found crispatus, and we found and most of it, crispatus and gasseri was quite common in the first weeks. But then, as time went on, rhamnosus became the dominant one because it was actually seen to be dominant during breastfeeding, the rhamnosus. But we found gasseri and we found crispatus early, but we didn't look for enos. You know, we had to grow them on blood agar plates, I think, in order to find them. But some enos, I think, was found anyway by sequencing later on. But early on, then other lactobacilli will take over during breastfeeding, like rhamnosus. And interestingly, on weaning, the rhamnosus went down significantly, and others came up, like Paracasiae, Plantarum, Delbrueckii, the ones you eat. So it really shows, and yeah. One thing that can have an opinion here, I think that we are very good at breastfeeding in Sweden, and I think it's, of course, very good, but we are also very afraid of giving anything else early on, and I think that might increase the diversity, the early diversity, just like I think the pacifier did, but they didn't look at diversity in this study in the gut, so we can't be sure. But what they saw was that the mouse flora in the kids that got the pacifier cleaned this way was different from the other kids' flora. But nothing was looked at in the fecal flora. But I think that if you should have a diversity, you have to get something into your guts too. Different things.
Katalin Kemerel, University of Cambridge. I would like to ask you a question which you already might have addressed. Why does E. coli has such a broad effect? Why do you see all this? Because this is not one effect only in one direction, it's in several directions. What happens there? Do you know anything about the mechanistic details behind all this effect?
No. I think that it's the same, it might be the same thing that you see in this metabolic syndrome discussions, where you have the endotoxin increased, perhaps, by the fact that you have more enterobacteriase there and also more fats at the same time. We didn't test E. coli in the low-fat setting, you know.
And no other bacterial strains as well, isn't it? It is only E. coli that you looked at all this.
Yeah, we have another study where we have looked at Lactobacillus plantarum and compared, but that was not really comparable. But we saw in the long run, when we were continuing this study, where we started in the pups and followed the rats till they became 6 months, actually. And some of them were given E. coli, others were given Lactobacillus plantarum this way. And then the weights of the E. coli was higher than the Lactobacillus plantarum.
Thank you.
Jim Geddard. The children in your first study are almost old enough to start thinking about the possibility of investigating childhood obesity.
Sorry, I didn't get what
The children in your first study are now almost old enough that you could study childhood obesity.
Yeah.
Have you thought about doing that?
No.
And have you thought about the potential problem of confounding by antibiotic exposure, which might also been related to the neonatal microbiota?
Absolutely. No, we haven't followed these children at all. We were not allowed to do that, to follow. We were not asking to do that, actually. But we have done a study in preschool children, 4 to 5 years old. And also there, we saw that normally weighted children had significantly lesser Enterobacteriaceae in their feces than had the overweighted and obese 5-year-old children.
One more question?
Yes, so just quickly, obviously one of the things that may influence the diversity as well as the specificity of what we might be finding in the gut flora will relate to what's happening in the uptake. And from what we know about, say, infections of Helicobacter and how that might influence gastric acid, that from a very young age, where we know that there are periods of hyperphagia lasting for up to a year, have you been able to look at that in any of your studies, or if not, are you aware of it? Because I'm not aware of any data on that.
No, I wasn't. No, I'm sorry. No.
One more very quick one if anyone wants one. Okay, in that case, thank you very much.